this is a problem that is shared among the world. 80% of death, 17 million people and more in the world, die because of cardiovascular disorders in low and middle income countries. So we are facing a situation in which in the big metropolitan cities of Africa and Asia, people die more of these conditions or of diabetes, metabolic syndrome than AIDS, HIV, malaria, and hepatitis C, tuberculosis all put together. And just to give you an idea of what is the impact of these diseases, look at this mail. This mail was addressed to our Director for External Relations, and it is from the African Academy of Sciences, which says that is organizing a workshop on capacity building in cell biology and regenerative medicine to take place in Nairobi, Kenya. The purpose of this meeting is to enhance the capacity of young scientists, African scientists in the area of cell therapy and regenerative medicine as to build capacity in this field. So they want people that can improve health in areas like muscle and bone loss, corneal regeneration, spinal cord injuries, myocardial infarction, and so on. So have you ever thought that these might become prevalent problems for the African continent only 10 years ago? I mean, that really surpassed my imagination. And an interesting issue is that, last time I told you that after myocardial infarction, basically you have a portion of the heart that dies. Now, in the Western countries, if a person has a myocardial infarction, so a sudden closure of a coronary artery, he or she is brought rapidly to the emergency room, and then to the catheter lab, cardiology catheter lab, and then the interventional cardiologist inserts a catheter up to the femoral artery, up to the heart, into the coronary artery, visualizes the clog that has closed the artery, and lies the clog and opens up the coronary artery. The person has a chance of almost 98% of surviving after myocardial infarction, so very, very high. But whatever is lost is lost. And a portion of myocardium that has undergone death never regenerates. And so it is substituted by a scar. And after a scar, the heart has less contractile power and remodels over time, by which it becomes larger, it thins, and it has problems in contracting. And this is a condition that is known as heart failure. So this is a table that a slide that summarizes the drugs that are available for heart failure. These are it's taken from the Mayo Clinic simply to signify which is the best treatment in the world. But this is the same treatment that you could have in Trieste or in your favorite city. So basically, there are 6 classes of drug, and then I played a bit at looking when these drugs appear in the medical literature. So basically, digitalis and diuretics dates back to 1910s or 1920s, but there is indication that digitalis as a plant was used even by the old Egyptians. Then in the 60s, the aldosterone antagonist. In the 70s, 2 classes of drugs that really made a change in the treatment and management of heart failure patients. So these are the ACE inhibitors and the beta blockers. And then in the 90s, the last drug, the sartans, so the angiotensin II receptor blockers. So this means that from the mid-90s to today, neither the pharmaceutical industry nor the academy, the universities, have been able to develop new drugs for this condition. This also means that the person with heart failure who is admitted to an hospital today is basically treated with the same drug combination he was treated 20 years ago. This gives you the burden of the problem, and this is the reason why we need definitely new therapeutic agents, new therapeutic strategies. Pharmaceutical industries are very good at developing small molecules, small chemical molecules, but it is very difficult to conceive that a small molecule can block degeneration of an organ, or after myocardial infarction, can induce regeneration. Small molecules are much more suited to block the action of enzymes, for example, inserting the pocket of enzymes. And very difficult to conceive that they trigger a very complex biological program such as regeneration of an organ. And so this is why the overall idea and the main theme of my laboratory is to find new biological drugs that could prevent degeneration, of this I told you last time, and induce regeneration. These new biological drugs could be genes. And when I speak about genes, I don't mean necessarily protein coding genes, but I speak of any nucleic acid, so an oligonucleotide of DNA, an RNA, oligonucleotide, an anti-RNA molecule. So everything that is based on a nucleic acid, or it could be a protein, or it could be a cell. So a cell that can be expanded in culture, injected in the heart to regenerate the heart, or even directly injected in the heart with the hope that expansion takes place in vivo. So last time I told you about a lot of how we try to find new proteins that can be used therapeutically. And what we do today is to tell you what we are trying more ambitiously to do to trigger regeneration. So the question here is to find new drugs that might trigger cardiac regeneration. So one interesting problem is the following, that is, but why does the heart don't regenerate? So there are other organs like the skin, the bone marrow, and the gut that physiologically regenerate. So why does the heart doesn't regenerate? So an interesting question is, when does cardiomyocyte proliferation stop? Because obviously in the prenatal life, the heart forms and enlarges, so there must be cardiomyocyte proliferation. Well, if you take a neonate mouse, so a mouse immediately at birth, you inject a thymidine analog that can be visualized by microscopy, for example, BRDU, bromodesoxyuridine. This intercalates into DNA when cells replicate, and so you can mark all dividing cells. You see this BRDU incorporation by microscopy as this, in this case, these red dots here. And if you do this in a neonatal mouse, you will see that more than 35% of cardiomyocytes are actively replicating. And this is the same as the embryonic life. So all through the embryonic life, up to birth, immediately after birth, the vast majority of cardiomyocytes are actively cycling cells, so they replicate. And in fact, there is work by which if you damage the heart immediately after birth, in the first week of life, there is complete regeneration, so no scar formation. So the heart is completely regenerated. And you can mimic this also in culture. So you can take neonatal cardiomyocytes, put them in culture, and you see that a proportion of these cardiomyocytes proliferate. For example, these are cardiomyocytes, and they are real cardiomyocytes because you can stain them with an antibody against alpha-actinin, which is a late differentiation marker. It marks the contractile apparatus. You see these are assembled cardiomyocytes with an assembled contractile apparatus, however, which express cyclin A, which is a G1S cyclin. So these are completed in the cell cycle. They incorporate bromodesoxyuridine, so BRDU, and they even show mitotic figures. So this is a mitotic figure in a cell which is green, so it has expression of the late differentiation marker. These are real cardiomyocytes that replicate. And as I told you before, if you take a neonatal heart and you induce damage to the apex, this is in the mouse, so the mouse survives, and you see that there is a tremendous trigger to proliferation. So this is a normal proliferation at birth. You see a considerable number of cardiomyocytes, but if you induce damage to the apex, you see that there is a stimulus to proliferate to these cells, and there is a complete reconstitution of the heart integrity. So there is the capacity to regenerate completely after damage that lasts about 1 week after birth. What happens that blocks cardiomyocyte proliferation? Fantastic and fascinating question. Nobody knows. There is really no clue on that. There are 3 important things that happen at birth. First, that the heart becomes an organ that is flooded with oxygen. So before birth, the heart is in the womb of the mother, in the fetus in the womb of the mother, and it is a very peripheral organ. So it's an organ where blood that comes is blood that has lost a lot of oxygen, so it is very venous. Immediately after birth, the heart receives blood immediately directly from the lungs. So there is a flood of oxygen. And this is one possibility that could be sensed by cardiomyocytes and by which they stop proliferation. This is a paper that was published last year. We contributed by Hesham Sadek. We contributed with a series of experiments. And basically, the idea of this paper is that it is the oxygen that blocks cardiomyocyte proliferation. And in fact, if you look at normal cardiomyocytes, immediately after birth, there is some proliferation. If you give H2O2 as a prototype for reactive oxygen species, you block completely proliferation. And you can extend instead proliferation after H2O2 if you give antioxidant compounds like sodium N-acetylcysteine. So the idea here is that this enormous flux of oxygens triggers mitochondria to burn some of this oxygen, not to produce ATP, but to produce reactive oxygen species. And so this oxidative burst is the one that signals to the cell to stop proliferating. And this is certainly a component. Another idea, which has never been proven experimentally, but makes a lot of sense, is that during the embryonic life, the heart starts pumping immediately. It's one of the first organs that forms in the embryo, and it starts pumping. However, all the circulation in the mother and the fetus is sustained by the heart of the mother. So the heart pumps, but it doesn't pump against a gradient. It pumps following a gradient, and so basically it has not much pressure applied. Immediately after birth, the heart faces the problem of having to sustain the whole circulation of the fetus, and so it has a tremendous hydrostatic gradient to pump against. And so this could be an important signal that stops proliferation and starts another biological program that is the program for hypertrophy. A heart in a newborn is very small, a heart in an adult is big. And the difference is not proliferation of cells, but it's simply increasing the cytoplasm of cell, something that in biology is called hypertrophy. So basically there is a program for physiological hypertrophy that enlarges the heart from birth to adulthood, and this program could be, again, triggered by pumping against the pressure, and this same program could also stop proliferation. If you need to hypertrophize, then you also cannot divide. And the third difference is a metabolic adaptation. So basically, before the heart, the fact that there is no need to sustain the circulation means that you have some metabolic needs which are very different after you are born. So this metabolic adaptation could also be a signal. There is really no proof that links this and makes also a bit less of sense that this could be the main signal. Whatever the reason, you understand that there is a biological program. And so the question is here, can we modify this biological program somehow? Difficult to do with chemical molecules, we thought. Let's see what we can do with biological molecules. And you know that in the cells, besides our protein coding genes, there are a couple of thousands of genes that code for microRNAs, which are small molecules that are endowed with the capacity of changing biological programs, because each of the microRNAs has a capacity to target tens or hundreds of different messenger RNAs and simultaneously downregulate all of these. And they have been selected through evolution to change the properties of the cells, to change even the fate of the cells. And so the question here for us was, can we identify, are there inside the microRNAs that are encoded by the human genome, some microRNA that could trigger cardiomyocyte proliferation, so it could be used for cardiac regeneration? So in Trieste, we have a facility for high throughput screening, and which is based on a robot, and we have libraries corresponding to whole genome siRNAs, anti-siRNAs, and whole genome microRNAs. So a very, very talented scientist from Portugal, Ana Eulalio, 3 years ago, started a very simple project. So basically, She took neonatal cardiomyocytes to provide cardiomyocytes with still a limited capacity of proliferation, that immediately after birth. She plated these cardiomyocytes in 96 well plates, and to each well, she added a different human microRNA. So she did a high-throughput screening experiment with about 1,000 microRNAs, and to measure proliferation, she added EDU, which is similar to BRDU, to measure incorporation. She waited 72 hours, and then she screened for proliferation. The experiment was made by high-content microscopy, which is a very, very powerful technique by which you have a microscope, an automated microscope that picks up a picture of the cells. In this case, the cells were cardiomyocytes. She added bromodesoxyuridine, so the nuclei that are replicated are in pink, and all the nuclei are stained in blue with hex. And you can see that in these pictures, there are also some nuclei that are replicated that are outside cardiomyocytes. And this is a good internal control because all cardiomyocyte neonatal primary cultures are contaminated 5% of fibroblasts, and so you can immediately check also proliferation of these cells. Then the microscope transfers the image to a software. The software calculates the contour of each cell, it identifies the nuclei, transforms the picture of the proliferating cardiomyocyte nuclei from pink to yellow, and simply gives you a number of the percentage of yellow dots out of the total cardiomyocyte nuclei. So this is a typical control. There are about 10–12% of cardiomyocytes still physiologically replicating at birth. If you do this experiment after 1 week, this is absolutely zero. And so she screened, and she found a series of microRNA, even to our surprise, that are very, very powerful in driving proliferation of the cells. You see here, for example, one of the most effective microRNAs, microRNA 593P. You see how many yellow dots you have here. So there is almost 50% of the cells that have incorporated BRDU. There are 40 microRNAs out of those we screen that increase proliferation very significantly of both mice and rat cardiomyocytes. These microRNA are human origin. We did the screening with the human microRNAs because we wanted something that had a chance to work also in humans. It's not true that microRNAs are so conserved across species. The seed sequencing might be conserved, but the actual action is not. So we wanted a reagent that might work in human cells. Some of these are really powerful. This is another of the powerful ones. You see, just to give you an idea, this is a plate of cardiomyocytes, neonatal cardiomyocytes treated with a C. elegans microRNA control 6 days after transfection. And these are the same cardiomyocytes, however, treated with this pro-proliferative microRNA. You see that the plate is full of cardiomyocytes. You don't need to do much molecular biology. You see that these cells keep proliferating. And you can also inject these microRNAs in vivo. This is made again in neonatal hearts. So on the left side, you see a control heart. After 7 days from injection of a control microRNA, this is the left ventricle, this is the right ventricle, and this is the pulmonary and the artery and the aorta. In green, you stain cardiomyocytes. And these are two hearts injected with two of the pro-proliferative microRNA, 590 or 119AA. You see that these ventricles are much thicker. And these are much thicker, and I remove the green. Simply, I don't know if you can see, but trust me if you can't. Simply because they have many more cells. This is EDU incorporation in vivo. You see these ventricles are filled of replicating cells. So basically, they are thicker because they have more proliferation. And if you look at a higher resolution, you see that there are a lot of cardiomyocytes in vivo which are perfectly embedded into the normal structure of the heart. However, their nucleus have proliferated in the last 48 hours because it has incorporated BRDU. Now, as you remember from my talk last time, we are somehow have a lot of experience with AV vectors. So for us, it was very easy to construct two AV vectors expressing the two most effective microRNAs. And an AV vector is very efficient because it goes into the heart, and then the microRNA gene simply expresses a precursor microRNA, which then undergoes the same process as endogenous microRNA. So it's processed by the microprocessor and then exported by exporting 5, and further processed, incorporated in RISC, and it does its job. And if you do this, you can check expression at longer times. So these are 12 days after birth, and you see that these hearts are really very big. I still remember the day when the postdoc who did this experiment came to my office with 2 tubes. There was 1 heart per tube, and she asked me, guess which is the one which received the control and which is the one that received the microRNA. They are very bigger hearts. Now, bigger hearts for cardiologists are not a good thing, because cardiologists tend to interpret the heart that becomes bigger as an attempt to recover a function that is lost. So typically, after a heart loses a portion of its contractile apparatus, even after myocardial infarction, when there is aortic stenosis, the heart tends to enlarge. But enlargement, in that case, is enlargement of the cytoplasm, so further hypertrophy, and hypertrophy can be bad hypertrophy leading to heart failure. However, here there is not hypertrophy. There are more cells, so it is hyperplasia. It's like taking a heart from a bigger animal with a bigger chest and put this into a chest of a smaller animal. And the interesting point of these microRNAs is that they are able to drive cardiac regeneration. So this is an experiment made by echocardiography. So basically, we induce infarction in mice by ligating the coronary artery. Immediately after infarction, we inject an AAV vector expressing the 2 most effective microRNA, 590 or 198P. And then we follow the functionality of this heart for 2 months. And this is a major parameter to measure functionality of the heart, which is ejection fraction. Ejection fraction, I think I told you already, is the percentage of blood that is pumped out of the heart at every heartbeat. In mice, this is the normal level, so a bit less than 60. In human is 70. If you damage the heart pumps less, so the percentage of blood that is pumped out is lower. And this is exactly what you see after myocardial infarction. You see this black bar, these white bars. You see that 12 days after myocardial infarction is less than 50, and then it progressively goes down. And this is heart failure. This is what happens in humans. If a human reaches less than 30%, 25%, then his only option now is myocardial heart transplantation. So there is no other possibility of curing this, definitely this patient. However, if the same heart you deliver the AAV of the microRNAs, then cardiac function is preserved over time, even after 60 months. And at the end of the procedure, you can check myocardial infarction, and basically what you see is that you have had a regeneration. So these are 2 hearts from a heart from 2 animals with a control, one sacrificed at 12 days, the other 60 days. You see that there is a big scar that had to be formed. So basically, this is the left ventricle, and here there should be muscle here. There should be muscle as here. Instead, there is this fibrotic tissue. So the whole anterior ventricular wall has been substituted by a scar. A human would never live with such a big scar. Rats and mice, as in this case, are much more resistant to this. The animal that received the AAV vector, you see the scar, is very significantly smaller because the tissue has regenerated. You can also quantitate this. So that was obviously very exciting because it was the first demonstration that you can regenerate the heart in vivo. But obviously this story opened up a very large number of questions. And in the cardiac field, as in several other fields, there is the big question of if you want to drive regeneration, which are the cells that are responsible? And we have witnessed for more than 10, 15 years all this huge saga of stem cells. So there have been a number of laboratories, and there are still a number of laboratories that believe that organ regeneration in adult animals must go through stem cell, and that several organs, including the heart, might contain some stem cell that upon proper stimulation can expand and then differentiate into the origin of into the cells of that specific organ. So for us, an important question was to understand which are the real cells that are the target in vivo of these microRNAs. So we did a complicated experiment, but conceptually very simple. So basically, we crossed 2 mice, and one mouse contained a flox GFP allele. Flox GFP allele, it means that it has the GFP protein between two, interrupted by 2 flox sites, and so basically it cannot be expressed. And this mouse was crossed with another mouse which contained the Cre protein, which binds to the LOXP sites, under the control of the alpha-myosin heavy chain promoter. And this is a promoter that is active only in cardiomyocytes. And this Cre protein is sensitive to tamoxifen, so it's not normally active, but you can activate it simply by giving to the animal tamoxifen. So you cross these animals, the result of the crosses still have their heart, which is not green, and all the other organs which are not green, but if you give tamoxifen, then this is expressed, and this is activated, and then it binds to the flox site on GFP, and the heart progressively becomes green. More than 80% of the cardiomyocytes in the heart become green. Should be 100%, but there is not, in these kind of genetic systems, there is never 100% efficiency. This is what is called a fate mapping experiments. So basically, we are mapping the cardiomyocytes, and we are at this point activating expression in adult animals only of cardiomyocytes. So all the heart is green. And then on these animals, we did infarction and injected the microRNAs and provided the EDU to mark the proliferating cells. So here the question was very simple. If the proliferating cells, so those that incorporate BRDU, are green, it means that it is the cardiomyocytes that were there that start proliferating. If the BRDU positive cells were black, it means that there are stem cells or other cell types, but non-cardiomyocytes, that kept proliferating, and these are the ones responsible for degeneration. The result was that all the proliferating cells were green. So it is really the cardiomyocytes that are the target for our microRNAs. You see they are the example of BRDU positive cells, and you see that the cytoplasm of these cells is always green. These are other examples. You see clearly this is a previously existing cardiomyocyte that have incorporated BRDU, so bona fide it has divided. So we thought, well, nice, but these cardiomyocytes were from an adult mouse, and people for 40 years have tried to have adult cardiomyocytes proliferating culture. They always failed. Let's see if our microRNAs are really capable to work also on adult cells. And to our surprise, we discovered that they do. And basically, if you take adult cardiomyocytes, you can put them in culture, and you treat them with the microRNAs, and you see that with a single exception, the top microRNAs that work in vivo, they also work in cultured adult cardiomyocytes. You see these cardiomyocytes that start incorporating BRDU, beautiful picture. See these monstrous, very big cells with an enormous cytoplasm with an assembled contractile apparatus that still incorporate BRDU and divide. And this reminds us on what was shown to happen in 2010 by the Belmonte laboratory working in zebrafish. Zebrafish and salamander are species where you can damage the heart and several other organs, and there is complete regeneration. And also there, people have thought for several years that there are stem cells that stay somewhere in the body that goes to the heart, and they're responsible for the regeneration. All false. What this paper shows is that in the case of the heart in zebrafish, it is the pre-existing cardiomyocytes that start dividing and keep dividing until in about 1 month, they completely regenerate the heart. And so basically what our microRNA do is to recapitulate what zebrafish does. And the difference between zebrafish and humans is that in zebrafish, this program for regeneration is maintained active all through the adult life. While in mammals, in mice, and in us, in humans, the program stops in the first week after birth. So for some reasons, we have this program active during embryogenesis and the first week of life, and then this is stopped. The reason could be evolutionarily explained because mammals need the heart absolutely for maintenance of the whole organism. If the heart doesn't work, I mean, the animal is dead, we are dead. And so basically, evolutionarily, there is a need to have a program for repair of scar or repair of damages to the heart, which is the fastest as possible. And the fastest program for repair is inflammation that triggers then fibrosis, collagen deposition, and scar formation. This is very rapid as a healing system. And since the evolutionary pressure is to keep the animal alive, this is what has been evolved in mammals. Whatever the reason is, these microRNAs are really active in adult cells. These are other experiments. This is a marker for proliferation. You see these, again, these giant cardiomyocytes that never replicate in normal conditions that are pushed to enter the cell cycle. You can even find in adult cardiomyocytes some mitotic figures, like, for example, this one. You see even the chromosomes start dividing, so which have never seen before. So it is the actual cardiomyocyte, the pre-existing cardiomyocyte, they divide, and so another question that is obviously paramount is what is the mechanism? How do these microRNA work? And so the first experiment we did was a very simple modeling experiment. So we said, okay, are these microRNA present in adult cells, adult cardiomyocytes that don't divide spontaneously, and in embryonic cardiomyocytes they do divide spontaneously? And so we checked by a microarray whether these microRNAs are expressed. And what we discovered is that several of these microRNAs are highly expressed in neonatal cells, so immediately after birth, and so we presume also in the embryonic life, but are downregulated in the adult. So we believe that by giving them back in the adult, we simply push the cell back to a prenatal state when physiologically these cardiomyocytes with these microRNAs were active. And so we wanted to understand what was the mechanism, and so we did an experiment by which we took mouse cardiomyocytes, put them in culture, and then treated these cardiomyocytes with the two, again, most effective microRNA, 593P, 98.3P. And then after 48 hours, we recovered the RNA from these cells and did next generation sequencing. So we did an RNA-seq experiment. And obviously, the idea is that I have a microRNA which expresses its own mRNA. I have a cardiomyocyte that expresses its own mRNAs. I give a single microRNA. I expect that this microRNA will target tens or hundreds of different genes, so they should be downregulated. If I do RNA-seq at this point, I will find all the direct targets in addition to the indirect targets. And basically, we found that there were 640 downregulated genes. We immediately look at which are the downregulated genes by the two microRNAs, and to our surprise at the beginning, but then if you rationalize it, not so surprising, we found that among the downregulated genes, there was a large prevalence of genes that are responsible for the formation of the contractile apparatus. So basically, the myomesins. There was even Titin. Titin is the biggest protein encoded by our genome. It's one of the stiffest. So it's a rod-shaped protein that marks half of the sarcomere. If you think, it's difficult to think that a big structure, cardiomyocytes, can replicate without at least downregulating the levels of expression of these big proteins. And so we asked what might be the effect of the downregulation of these individual proteins, the contractile apparatus, on proliferation. So out of the 641 genes that were downregulated, we obtained 600 siRNA. Again, this is a high-throughput experiment. And we asked what is the effect on proliferation of knocking down individually each of these 600 genes. So if you knock down genes, for example, of the contractile apparatus, do you see replication by itself without a microRNA? And this is the result of this experiment. So basically, this is a proliferation of mouse cardiomyocytes. It's lower than rat, as I showed you before. It's about 5%. This is a proliferation imparted by this microRNA here, 199A3P. And this is the level of proliferation that is imparted by treating the cells only with one specific siRNA. And these are the siRNA corresponding to all these downregulated genes. So it means that the total effect of the microRNA is the sum of these effects here. And again, among these effective siRNAs, we found the siRNAs with proteins of the contractile apparatus. For example, there was cofilin 2, which is an inhibitor of actin polymerization. There was ancar-D52. There was Titin itself. So if you knock down Titin by itself, look at this experiment here. This is a proliferation in normal condition. You give the microRNA, you see this proliferation for 199 and this for 590. If you knock down Titin by itself, where is it? Here, you see this level of proliferation, so still significantly higher than this one. But for example, if you mix Titin with confilin 2 and ancar-D52, so these three, you see proliferation which is equal or even higher than the microRNA. So we believe that a vast part of the activity of the microRNA is due to the capacity of down-regulating the contractile apparatus. This is why I believe that the stimulus to hypertrophy is very well linked to the stimulus, the inhibition of the stimulus for proliferation. Hypertrophy means building up a huge and large contractile apparatus. So if you block this building up by an siRNA against the relevant genes, then the cell could have a default program for proliferation. Apart from the mechanism, obviously here, the question, which is probably more relevant even, is at what extent can we use this information and these reagents for developing new therapies? So after the paper was published a couple of years ago, there was a big fuss in both the scientific literature and in the popular media, because immediately people envisaged the possibility of developing new, very simple drugs based on microRNAs. And this is an editorial on the New England Journal of Medicine by a group in the Karolinska, which was very generous to our work. And basically, they draw a very simple diagram. So they say, you have a heart with an infarct. Well, simply what you do in the infarct border zone, you inject some of these microRNAs, and these microRNAs can trigger cardiomyocyte proliferation, so cardiac regeneration. Obviously, very easy to draw, a bit more difficult to do, but this is exactly in principle what we would like. What we would like to do is to have an ideal clinical scenario in which a patient has a myocardial infarction. He is brought to the cath lab for angioplasty. He has his coronary vessels reopened, and after that there is a portion of myocardial tissue which is lost. There is a lot of inflammation. It is known that this inflammation releases factors that trigger fibroblast proliferation and collagen deposition after 3, 4, 5 days. And the idea is to wait 3, 4 days, and at that point, with the same catheter, inject a bolus of these microRNAs that should enter the different cells and trigger proliferation. So if instead of allowing proliferation of fibroblasts, we trigger a bit proliferation of cardiomyocytes, then we would at least restore a portion of the myocardium. Obviously, we have vectors to do this, which are very, very efficient. So this could be done. But honestly, even if I would have a myocardial infarction, I would not an AV vector with a pro-proliferative microRNA be injected in my heart, because I told you last time that an AV vector expresses any gene forever, basically, for the whole life of animals. We have experiments with collaborators in the US, in pigs and dogs, and they have animals injected 6 or 7 years ago, they still express the protein. So because this vector go to post-mitotic cells and it keep expressing for the whole life of animals, we see this routinely for 2-3 years with mice and rats. So basically here, the idea would be to have a synthetic microRNA that lasts for a relatively short period, but enough to trigger some proliferation. And the good news in this respect is that even normal microRNA mimics, so the one that you buy from commercial sources, could do the job. This is an experiment in which basically we induced myocardial infarction and injected the microRNAs corresponding to 99 and 590, so the same two most effective. But this time, the microRNAs were not in AV vectors. They were just synthetic microRNAs, simply mixed with some cationic lipids. Again, something very commercial. This was the RNAi max preparation for transfection of microRNAs. And this is measurement of the persistence of the microRNAs in the heart. You see that in 24 hours after injection, there is a lot. This is a fold the endogenous levels, which are very low. So there is a lot, and it goes down very, very rapidly. But at 5 days, there is still 25 times more than the endogenous. For this microRNA, 250 times more than endogenous. And so the question is this time and levels sufficient to drive cardiac repair? And functionally, the answer, it appears that it is. So this is a left ventricular ejection fraction measured up to 2 months in mice. And you see that cardiac function is very significantly increased compared to control in the hearts that have received this synthetic microRNA, only 1 bolus. Consider that in humans, one can repeat these catheteries, for example, after 1 week or after 2 weeks. So we are very, very excited by these findings, and now we are trying how to stabilize better these microRNAs, how to mix with more effective polymers, but to keep the microRNAs always as synthetic molecules and see if we can develop a new medicine based on this small nucleic acid. I think that from the, and this is my last slide, that from the conceptual point of view, I'm on time, no, Glauco? Okay. That from the conceptual point of view, I think that this kind of work provides an important contribution, because so far we have heard all around the different fields of degeneration or regeneration the importance of stem cells. And for the heart, people first have claimed that there are bone marrow stem cells that can become cardiomyocyte, completely untrue. Then that mesenchymal stem cells from the bone marrow can become cardiomyocyte, completely untrue. Then that the heart might contain cells, stem cells that can become cardiomyocyte. Still under question, but I would say that 90% of people are not in favor of this possibility. Certainly, there are stem cells from embryos that can be grown in culture, and they can become cardiomyocyte and can be transplanted. But this is obviously a very difficult kind of medicine, because think of a disease which affects 1 person out of 3, and of which 80% are in developing countries. Think at treating this disease by having large batches of embryonic stem cells that are grown in culture, convinced to become cardiomyocytes, and then the cardiomyocytes are injected in the heart. This would be a medicine for 0.000 something percent of these people. Instead, the idea that you can trigger proliferation of already existing cardiomyocyte cells by, for example, microRNAs, would allow these microRNAs to be put in vials, and these vials can be injected in the heart by any interventional cardiologist all around the world. Cardiac catheterism is the most executed interventional procedures all around the world. So there are a lot of cath labs that do this, and if there is something to be injected, then this could really be a new paradigm for therapy. If it will work with microRNA, we will see. We are pursuing this and also trying to find other microRNAs and anti-microRNAs. We will see, but I think that is an exciting moment to live now. A lot of people are involved in this work, so I won't mention anything. But I have to say that I'm very, very happy and privileged to have the opportunity of speaking about this work that I find fascinating. Why I have a fantastic team in the laboratory who does all this work here. This is all work performed at the ICGEB here in Trieste, and I'm very, very happy to share this with you. If you have any question, I'm very pleased to.